Hi, my name is Molly Bright. I'm from Northwestern University in Chicago in the US, and this is the first talk that's part of a day-long educational course at OHBM 2023 in Montreal. The course is about physiologic fMRI signals. Are they friend or foe? And how and why you should measure and model and account for physiology in your neuroimaging data. The core content of this talk, which is the introductory talk for the course, the content really hasn't changed from last year very much. Although I will have slightly updated slides, the main material is the same, and thus the rest of this recording will be borrowed from the first offering of this course in 2022. So the background may say Glasgow, but hopefully you can still get all of the salient information about physiological recordings. Hi, my name is Molly Bright. I'm from Northwestern University, and I'm here to remind you that the brain you're very interested in studying with an fMRI scan is inside of a human being. So with all of us at the Organization for Human Brain Mapping, we're interested in understanding how the brain is functioning, and typically we're trying to use functional MRI to study neural activation or connectivity, and using this to explore correlates of behavior and cognition and very high-level things. But that brain is inside of a head that is attached to a human being. And there are many things that we need to think about when we start to scan that brain in that person. And a lot of them I think we're already quite comfortable at thinking about. So, did our participant recently eat a meal? Have they had caffeine in the last hour or so? Did they get a good night's sleep last night? Are they on any medications? What time of day are we scanning them? Are they very stressed? Do they exercise heavily today on their way to get to their scan? Do they exercise regularly in general? Are they a smoker? How heavy of a smoker are they? Are they experiencing anxiety, or are they uncomfortable like right now during the scan? Or, is that a chronic thing for them? And we're used to thinking about these as potentially influencing what we're seeing in the scanner. And so either we try to have self-reporting and write down and acknowledge some of these things and this physiologic variability in our participants, and we can try to mitigate these things as well. So we can schedule scans for a certain time of day and ask people to refrain from eating or drinking ahead of time. But ultimately, there's a lot still going on physiologically that's varying during the scan itself. So we can't turn this off if our human being is in there alive and thriving. So what can we do? We can measure it, we can understand it, we can model it, and we can incorporate it into our fMRI experiment. So in this talk, I will be asking what else is happening in the human body that is hosting your brain. And I'll divide it up into three sections: the lungs, the heart, and kind of everything else. We'll discuss how these different things might impact imaging data, how we can monitor and record these things during imaging experiments, and, in terms of what models exist to connect what we record to what the fMRI brain signals are, that I'm not going to cover. That will be done by other people in the course. But I will make some general recommendations for how you could incorporate these things into your experiments. So first up, the lungs. Your lungs are inflating and deflating approximately 12 to 16 times per minute if you're breathing at rest. And in order to understand what that can do, I think we want to just clarify that there are two distinct things at play with your lungs. One is ventilation and one is respiration. And in casual conversation, we may just conflate the two terms, but they are different processes, and it's worth thinking about that. So what do I mean by ventilation? Well, that's the breathing bit, right? So that's inhalation and exhalation. And your brainstem is sending signals through the spinal cord and the phrenic nerve to cause your diaphragm to contract, and your lungs therefore expand. And then when the diaphragm relaxes, your lungs will, constrict as well with the weight of the cavity that they're in. And as these lungs are expanding and contracting, air is going in and out of your system. That is breathing, and that is what we call ventilation. So what is respiration then? That's all about gas exchange. So where is that gas exchange happening? I'm thinking about the thin membrane that is separating the gases in your blood inside of the pulmonary capillaries from the gases that are inside of your alveoli, deep inside of your lungs. And exchange of these gases across that membrane is external respiration. And that's what we're going to be talking about. For that exchange to happen, you need to have a pressure gradient in place. And oxygen is going to move from the air that you breathe in into your blood and attach onto the hemoglobin and get traveled throughout the body. And carbon dioxide is going to leave the blood and exchange into that gas in the alveoli and eventually be exhaled. Now, ventilation and respiration, although two distinct processes, are very, very connected. So changes in one can cause changes in the other. So how do these ventilation and respiration phenomena, how do they influence the fMRI signal? First thing that can happen is that breathing will actually cause your head to move in a very periodic manner. And this will primarily be in the pitch and Z translation directions. So this will cause misalignment from one volume to the next, but it can also lead to what we call spin history effects, where because of that movement, the signals in a given voxel are different for several acquisitions. So this is associated with ventilation, and you have an immediate effect plus a few time points afterwards due to that spin history. You can also have the position of your torso, so where your chest is inside of the scanner will influence the B0 main magnetic field that the scanner is creating. And this can lead to several artifacts, including like this apparent or pseudo head motion in the phase encode direction. And so this is also ventilation related, and it's immediate as well. You can also have changes in arterial blood gas levels. So your blood is holding oxygen and carbon dioxide, and these levels can change over time. And this is important because arterial CO2 can dramatically change your vascular tone. So as your arterial CO2 levels go up, your vessels will dilate, and as the CO2 levels go down, your vessels will constrict. Therefore, how much CO2 is in your blood will change vascular tone and blood flow, and that will directly influence this fMRI bold-weighted signal that you're acquiring. So this has more to do with respiration because we're talking about how blood gases are transferring between blood and your breath. And it's also a delayed effect because that change in blood gas levels has to travel to the tissue that you're imaging. It has to cause the vessels to change their tone and then drive that blood flow response. So it's a delayed effect. So how do we measure all of this? To measure ventilation, we can use some type of stretchy belt around your torso to estimate your chest position and how that changes over time. So where would you place that belt exactly? On this little schematic, I'm showing you 3 potential locations: the top of your chest, the bottom of your rib cage, and on your abdomen. And I think a lot of vendors and a lot of users of these belts will advocate for a different positioning. I'm not going to tell you what is best, because what is best is what works for your participant in your experiment. You should know that if you put the belt on somebody and it seems like a good position to capture their breathing, if you put it on when they're sitting and then they lie down supine on the scanner bed before they move into the scanner, a lot of times the belt position can shift, so you may need to adjust it. And also the breathing style of a person can change when they lie down and also as they relax as the scan evolves. So you may also begin to not capture the breathing patterns accurately as time goes on. So I think several people have advocated for employing multiple belts in order to fully capture ventilation and breathing of a participant at all times of the scan. And what are you looking for in the signal? So you see here a clear, periodic, almost sinusoidal shape of somebody breathing. And what you don't want to see is any clipping or saturation of the signals at the peaks and troughs of that time course. That would probably indicate that the belt that you've put on is too loose or too tight. Some of the belts are going to be an electronic sensor, some of the belts are going to be pneumatic, and they all have slightly different characteristics, but all of them can be MRI compatible and give you similar information about ventilation. So how can we measure respiration? Typically that's through something, usually a nasal cannula, which is a piece of soft plastic tubing with two prongs that sit inside of your nostrils, and typically it will loop around your ears and be tightened under your chin just to have it secure and in place. And this will allow us to measure the CO2 and the O2 levels of the gases that you're exhaling. And so it helps us access information about your respiration, among other things, such as metabolism. And this sampling of the gases that you exhale, that's a non-invasive surrogate for direct arterial sampling, which is kind of difficult and messy to do in the MRI environment. But in order for it to be a good surrogate, you need to be able to extract what we call end-tidal values from these data. So what are they? Here's a trace of CO2 as a person breathes out, and then in, and then out, and then in. There's very little CO2 in the air that you breathe in if it's just standard room air, so it's quite low, and as you exhale, because your body produces carbon dioxide, it goes up. And what we see here is at the end of the exhalation, I'm noting with this orange circle, that is the end tidal value. It's the very last bit of air that you exhale before you then take the next breath in. And this air is coming from maybe the deepest part of your lungs where we think it's most in equilibrium with your arterial blood gas levels. So it's our best estimate of arterial blood gas levels. So here are examples of good and bad CO2 recordings. On the top, you see a trace that has clear end tidal values, and I would say that's acceptable estimates. On the bottom, you see something that looks a lot more sinusoidal. You don't see that plateau and then a sharp drop as the person begins to inhale. So these are poor end tidal estimates. I would also note that you see high CO2 levels at inhalation in this particular data set. To me, this suggests that there's a lot of dispersion happening. You might have a very long sample line connecting the nasal cannula to the gas analyzer in the control room, and the vacuum settings might be too low. So you end up with a lot of dispersion of this exhaled gas that you're recording. You do need to calibrate these setups, and you need to have the identical setup that you'll use in your imaging experiment when you calibrate. So the same length of sample line, the same nasal cannula, and the same vacuum settings. But there are two big caveats with using a nasal cannula. And the first is that you are always going to entrain some room air into your measurement, right? It's not a sealed system. And that will influence the accuracy of your measurement of the end tidal values. It will be diluted by that room air you haven't trained. Secondly, you need your participant to breathe through their nose in order to get this measurement. So some may not be as compliant as you need them to be. So in those cases, there may be experiments where actually using a face mask to accurately capture exhaled gases and measure CO2 and oxygen levels, that might be a better, although understandably a bit less comfortable and pragmatic option. Okay, moving on to the heart. Your heart is pumping approximately 60 to 100 times per minute at all times if you are happy and healthy during that fMRI scan. And I want to just clarify that when we think about the cardiac influence on fMRI data, we're thinking about the heartbeat itself, but that is distinct from another topic that we do think about, which is the heart rate or your pulse, the frequency at which your heart is beating. So your heartbeat, this cardiac cycle, it involves ventricles contracting, and that's in systole, and then relaxing in diastole. And this periodic contraction will pump blood throughout your circulatory system, and its goal is to deliver oxygen and nutrients to the body through the bloodstream and remove waste products from the system. The rate at which your heart is pumping is actually a sinus rhythm set by pacemaker cells in the heart itself. But this is modified continuously by autonomic nervous innervation. So parasympathetic nerves can slow down the heart rate through the vagus nerve, for example, and sympathetic nerves can increase the heart rate. And both of these factors are constantly acting on the heart. And so your heart rate is in constant tension between this drive to slow it down and speed it up. Things like physical activity, metabolic byproducts, the status of your muscles and joints and tendons, and inputs from your limbic system can all impact your heart rate as well. So how does this beating of the heart influence fMRI data? Well, it creates a rapidly traveling pressure wave throughout the vascular system and anything that's nearby. So here's a video of very rapid MR images, and you can see the pulsation, the large vessels going up the brainstem, and you can see all of the ventricles above that are pulsating with that cardiac cycle. And so this causes what can be considered local movement artifacts. So within a voxel, the actual substance within that voxel is changing as things are pulsating and changing shape. So you'll have changes in many different parameters. You'll also have pulsatile flow effects. Any movement of the blood or other fluids flowing through plane will have some sort of spin history or T1 effect as well. And so what exactly is moving other than blood? Well, when your heart pumps and blood is sent into the brain, your brain is encased inside of the skull, which is a rigid fixed structure. It's not going to expand. So as the blood comes in, something has to come out. And this is known as the Monroe-Kelly doctrine. And what typically is the case is that as blood comes in with each heartbeat, CSF has to leave the intracranial space in order to maintain pressure and not basically have your head explode. And this thought process has actually moved into this very exciting field studying glymphatics or neurofluids and how pulsation and other types of periodic movement of fluid could be key, not just for getting nutrients to the tissue, but flushing waste out from the brain. And that could be particularly important in things like Alzheimer's disease. So that's a whole new field. I'm not going to touch it further than that, but you need to capture that heartbeat in order to study some of these really interesting directions. So how do we measure when the heart is beating? Typically, that's with photoplethysmography, or PPG. And this is an optical measure of blood volume changes in your periphery. So typically, in some type of device, an LED will illuminate your skin, and a sensor will record the light that's either transmitted through that skin or reflected from it. In the special case, you can actually have true pulse oximetry where you measure and quantify SpO2, so measure oxygenation levels. This requires multiple LEDs in your device, so in the red and infrared part of the spectrum. This is akin to NIRS. Most devices, however, are giving you more qualitative, relative changes in blood volume. And the device is usually a finger or an earlobe sensor just for ease of attaching it to a person. But depending on where you put that sensor, you're going to have a different travel time of a given pulse wave, and you may have different oxygenation dynamics and oxygenation levels. There are alternative ways to monitor and record the cardiac cycle other than a PPG. You could have a pressure-based sensor also on, say, the fingertip, and from that wave, you can actually do a pulse decomposition analysis to estimate blood pressure variations on a beat-to-beat level. You could also use ECG, so electrodes that are positioned on the skin by the heart. This is typically part of an MRI scanning setup, so it might be something you have ready access to as well. However, with all of these things, please note that typical fMRI temporal resolution does not actually resolve the cardiac cycle. So these effects are going to be aliased into lower frequencies. Typical TRs in fMRI data, even a second or so of a TR, if the cardiac cycle is also at about 1 hertz, you do not quite meet the Nyquist criteria. So you are going to be aliasing that pulsation into something low frequency. There are exceptions, however. So I'll give you an example here of MR encephalography or MREG data. This is a fast TR technique, and you can resolve the cardiac cycle if you're scanning every few hundred milliseconds. So here is a video where you can see that cardiac pulsation, and it's fully time resolved with this technique. Okay, so that's the heart. What about the rest of the body? And this is going to be a whistle-stop tour. So, your skin, your largest organ, and we can measure what's called skin conductance. If you look historically, it's also been called the galvanic skin response or electrodermal activity. And conductance through your skin changes, and it's going to be proportional to the number of sweat glands in that area of skin that are activated. So your sweat glands will be activated in a way that's associated with stress, arousal, or emotional excitement. At an extreme level, you could imagine a fight or flight situation where you're presented with a perceived threat. You're going to have to run away, and so your body proactively tries to cool you down. And that could be a very large effect in skin conductance. But just thoughts and emotions could trigger similar pathways in a smaller way. So when we're talking skin conductance, we're going to see some evoked things. So we can provide a stimulus and create an event-related skin conductance response, or SCR. We could also have unevoked SCRs. These are non-specific responses. And then we also see these spontaneous, very low frequency fluctuations in the tonic skin conductance level. So here's what that could look like in an experiment. In the beginning, you have somebody just reading instructions, very neutral. Then they begin to have some sort of gambling task with high-risk choices. And at the end, it's again a neutral questionnaire. So you can see that there's a change in the tonic skin conductance level from this neutral condition to a high-risk condition to, again, a neutral condition. And on top of that, you can see these phasic responses that are happening periodically. Those are the SCRs. And so skin conductance gives us access into the sympathetic nervous system. How do you measure it? Well, you got to clean your skin, apply a gel, and affix electrodes, and there doesn't appear to be really an established standard positioning. However, most things I've seen involve placing the electrodes on adjacent fingers, as shown in this diagram. You want to minimize hand movement, just like with the pulse recordings. So you want to support the arm and hand with cushions. You should test that you're sensitive to skin conductance responses, and you can do that by having a participant just take a deep breath to evoke that response. And you'll need to apply filtering and other signal processing techniques to really separate out the phasic from the tonic effects. You should be careful with the temperature and humidity of the scan environment, as well as the hydration and medications that the participant is on, which can influence skin conductance as well. What about the eyes? There are many MRI-compatible eye tracking systems, and the eyes are a window into lots of things about your physiology. So at the very least, an eye tracking system can tell you where you're focusing your attention, and that may matter if you have very complex visual stimuli being presented. But with these systems, you can also do pupillometry and measure the diameter of the pupil. And this diameter will respond to any emotionally charged stimulus, be it positive or negative valence. And your pupil size will also respond to changes in your cognitive load. So when you're working very hard and you have higher cognitive demand, your pupil size will get larger. But when you're thinking about this, you need to make sure you control light levels, because light levels are the biggest influencer of pupil size. So you can standardize the luminescence in your stimuli, for example. And there are several papers, and I'll show those in the references, indicating widespread negative visual and sensory motor signal changes in fMRI data associated with increased pupil diameter, as well as many signal changes associated with the change in pupil diameter over time. It may also be possible to estimate where the eyes are looking from the fMRI data themselves. So this is somewhat recent, but your eyes are moving. You can train somebody to look at certain parts of a stimulus in a certain order, and then use those data to train neural networks to then decipher, based on the signals you see in the eyeballs of your fMRI data set, which direction are the eyes looking at. And this is shown to be quite similar to camera-based eye tracking techniques. So very promising. Okay, moving lower, how about the gut? It's a really interesting emerging field using fMRI to study the gut-brain axis. So the gut is constantly producing a gastric rhythm. There are, just like the heart, there are pacemaker cells in the stomach to generate something at about 0.05 hertz, which already sounds kind of overlapping with many other interesting processes in the brain. And these occur even when the stomach is empty. So you can record them by putting electrodes on the skin. So this is called surface electrogastography, or EGG. And you can see in this figure here an example of where electrodes can be positioned. And papers have shown that there's significant coupling between this EGG signal in that low frequency band and bold fMRI signals in many brain regions, each one having a slightly different temporal delay. So this is an emerging field and could be a really interesting thing to add into your experiments. Okay, now for some general concepts that span all of the organs I've talked about. It's worth it, guys. I strongly encourage you to take the extra time and effort to incorporate high-quality physiological recordings into your experiment. You get multi-parametric, complementary insight into the function of the brain. And from that, you should also be able to derive more specific and hopefully more accurate measures of neural activity in your fMRI data. And although there are many data-driven techniques that are emerging that may mean you don't have to add a lot of extra recordings, some of them aren't quite ready for prime time, and particularly in pathology. So I think it's still very much important to get these recordings and help contribute towards the data-driven techniques. What equipment do you need? You need to have the triggers from your scanner so you know when the volumes were acquired. And basic physiological monitoring will involve a PPG, something to measure the pulse, and a belt to measure ventilation. A lot of times these come with your scanner, making it very easy, but you can also get third-party systems. You may need a nasal cannula and gas analyzer, a skin conductance system, an eye tracking camera, etc. And if you have these third-party systems, you'll need some type of analog to digital converter and recording software to record and synchronize things from multiple sources. And in terms of extra effort, you need to set up this equipment ahead of time. You need to position the devices on the participant once they're in the scan room. So you are paying for that scan time, and you need to use some of those minutes to actually place these things on the person. You'll have to do some quality control of your recordings in the first few minutes of the scan, typically during your localizer or a T1 anatomical. You may need to go in and reposition the devices if the quality isn't sufficient, and at the end you'll have to export and process the data. So tips for good recordings: please check the quality before your fMRI scan takes place, because it's really worth going in and repositioning if needed. You want to make sure your breathing trace from that belt is smooth and it's not saturating or clipping. Have someone take a deep breath in and out to make sure, and you can adjust the tightness. And, once they're lying supine, if you don't see a great signal, just watch how their torso is moving and just slightly reposition the belt to better capture that. If your expired gas trace does not have distinct end tidal values, make sure your vacuum's at a very high setting and try to make the sample line as short as possible. And you should see a very clear heartbeat above the noise floor from your cardiac PPG sensor or other. If you don't, you may need to adjust the tightness of the sensor. You may have a particularly cold hand, so you could put a blanket on top of it. And I know in my group, often we do a motor task involving the hands, and so there's too many movement artifacts on the pulse, so we can actually find an artery on the foot. I do encourage you to record continuously throughout the entire scan session. Some modeling that we do with these signals benefits from being able to shift the data back and forth in time. So having a bit of information from before and after a given fMRI scan period could be beneficial for that. And you may also want to add markers to your recording software to identify different experimental elements. Think about your trigger. Some signal models want to trigger every volume or every slice, multiband. Learn how to find slice timings in the header of your data and what that means for using that trigger in the processing of your phys data. Analog triggers may need a little bit of massaging in order to have a crisp delta function when you go into your processing. Save your data in a sensible format, and share your data when you share your fMRI data. And we're going to talk about all these things a bit later in the course. Always test when you have a new phys monitoring device. Always test if the signals you get are corrupted by the scanner noise. And always test if they're bringing noise into your MR images. So work with your imaging center, test for safety and for signal quality. Pay attention to the materials, the cable shielding, and how signals are actually going in and out of the scan room. And I encourage you to use the same experimental setup for every scan that you do, and even better, have everyone in your center use the same setup for every scan at your institution. This will really streamline the formatting of your data, the signal processing steps, and it will make it easier for other users at your institution to start incorporating these recordings into their own work. All right, In the last few minutes, some advanced recommendations. I encourage you to evoke a physiological change on purpose, because bigger effects are easier to capture in your data. It improves modeling of physiological phenomena. And one caveat, though, is that evoked and spontaneous physiological fluctuations are probably a little bit different. So here's a case study. In my lab, we do this very pragmatic hybrid protocol for studying vascular and neural function. So we have, in the beginning of the scan, we evoke larger breathing changes with these breath hold tasks, and then we have a very typical resting state paradigm afterwards. And we record the breathing with a belt and a nasal cannula. It has a bit of redundancy in case somebody does not breathe through their nose. We can still work with these data. And we do continuous recording, so we have information about breathing before and after the scan itself. So this protocol was developed by my postdoc, Dr. Rachel Stickland, and a lot of the data I'm going to show you on the next slide or two is from my PhD student, Christina Svolanek. So here is an example of phys recording from that hybrid fMRI protocol. We have the trigger, CO2 and O2 traces, a respiratory belt measuring chest position, and the pulse trace from a PPG sensor. And you can see it's very obvious the features of our protocol. You can see the 3 breath holds followed by spontaneous fluctuations of physiology at rest. There's no saturation of the belt signal, and we don't see any interruptions in the pulse, perhaps due to hand motion. If we zoom in on the breath hold, a few more things become apparent. We do have nice end tidal values, so big positive peaks at the plateau in the CO2 and at the end of the trough in the O2 trace. But there is a very large temporal offset of almost 2 breaths between the belt and the end tidal, or the exhaled nasal cannula gas recordings. And we expect that there is some delay in the gas changes. If we zoom in on a resting state period, however, we see nice clean waveforms, very periodic and repeatable, but we may not actually see the true offset between some of these recordings like we can in the breath hold. So this is why I say it's helpful to evoke a large change. It helps you understand the relative timing of all the different aspects of physiology. So in this type of acquisition, you can see here the changes in CO2 caused by the breath hold and then the resting state portion. These are end tidal values that we've extracted. And you can see the bold fMRI time series in gray matter, very strongly reflecting that physiological change. And from this, we can actually quantify aspects of vascular physiology. So here we get a measure of the amplitude of that response called cerebrovascular reactivity. And we can also measure the delay of that response because we have information before and after the scan, and we can really be sensitive to interesting pathology like we are here. And this information is just so complementary to what you then acquire as a traditional resting state fMRI scan. So with that, I will end, and if you want to have one take-home message from this talk, you might as well try it because phys data are pretty useful, and I think you should all be encouraged to attempt to add these recordings to your own experiments. Thank you.